Chapter 3, Nature of Antigens and the Major Histocompatibility Complex. That's actually called MHC. That's what you'll hear me talk about it from now on. So we'll be going over the factors that actually influence how the immune response is. What actually controls it. What are immunogens, which is really a better word really for antigens. What are the traits, what are the characteristics of one? We'll explain what an epitope is, what haptens are, what adjuvants are, the relationship of what the antigen is to the host, which as we're going to talk about it as dealing with humans, and what MHC is, and what its role is in the immune response. So what does affect the immune response in people? There's plenty of factors. One of the big ones is age, and it's the very young and the very old. So neonates, when they're born, their immune system's not yet completely developed yet. That's why breastfeeding is very important, for them to get antibodies in there in the colostrum, the very beginning part of the breast milk for that. Also, they don't have any, indigenous microflora, no bacteria in their GI tract. So, none of that's there, and that has to slowly be developed. On the other hand, older individuals have a very decreased immune response a lot of times. It's just because it happens with age. The body is slowly checking out, and the immune response is not that good. Now, that's not for all people, and then it changes for a different age. We're talking about 80, 90, 100, something like that. But some, it really doesn't really affect them overall. And of course, the health of the patient. Are they eating right? Are they getting their correct foods? Malnutrition does, cause problems, at any particular age. Also, fatigue. If the person is tired all the time, they're not getting sleep like they should, sleep deprivation, working too much. And then a big one, which a lot of people forget about, is stress. If you're under a tremendous amount of stress. Now, we're not talking about stress of typical daily kind of stress. We're talking about like a death in the family, and with COVID going on, a lot of stress has occurred. But, stress can be a big player. You've probably seen it, if you're under stress a lot, it seems like you get the sniffles, or you don't feel right, or something like the next day or so. So that's an easy thing that can cause the immune response to be decreased. Other factors that influence immune response is how we get these foreign things in to us. It can be either intravenous IV, intramuscular IM, intradermal, subcutaneous, just under the skin, or oral. If you look at your vaccinations, that's how we determine the best way to get it. So that's the things we look at that can possibly affect the immune response. Also, the dose. If it's just a little tiny bit, then that's probably won't be enough to cause us to elicit an immune response. Very large doses sometimes can induce tolerance, and we'll talk about that with allergy shots and stuff like that. But that's why it's so important to know how much people are exposed to. And then, of course, the genetic predisposition of the person. You've seen some people, they're allergic to everything. And then some people are allergic to nothing. Some people will get all kinds of stuff. Diseases, it's like everything that comes down the pike, they'll get it. And others that won't. So, what is the actual definition of immunogen versus antigen? Well, antigen, like we said, just means it's something foreign. It's different than us. In immunology, we always talk about self and non-self. So an antigen is actually a substance that the immune system can recognize. An immunogen is something that the immune system sees as being formed, but will stimulate an immune response. Now, antigen doesn't mean you'll produce antibodies, where if it's a true immunogen, it will definitely produce antibodies. So the traits of immunogens that make us influence the immune response there's quite a few of them. One of them is the actual size. And now we're talking about molecular size. So we're looking for a macromolecular size. So it has to be what's called 10,000 Daltons. But, you know, it's got to be big enough. We'll say that. Most immunogens you can see are greater than 100,000. So that's, you know, and when we get into another definition here, that'll play an important part. But it's got to be big enough. And the more different it is from the host, from us, the better chance of it eliciting an immune response, better chance of it being an immunogen. Proteins are actually the most effective immunogen, followed by polysaccharides, and that's actually due to the complexity, which we'll get here in a second. So when we talk about proteins, we look at the structures of them. So we've got the primary protein, the secondary, that's the alpha helix where it's pleated, the tertiary where we start getting more of a three-dimensional, and quaternary is multiple three-dimensional things. So the bigger it is, the better chance for us to elicit an immune response. So what part of this immunogen, this foreign thing, does the lymphocytes actually look at? I mean, you saw it. I mean, it's a big protein. So what does it look at? So it looks at a particular part on that foreign thing, and that's actually called the epitope. So there are different types of epitopes. There's ones that are linear, and there's ones that are conformational. In other words, like a three-dimensional kind of look to it. So linear means sort of like a straight line. I mean, you can see it here. The picture's here. You can see linear here and conformational here. So actually, they're showing you in the green here that is actually a B cell. This little Y-looking purple thing sticking off, it's an antibody, and you can see they've blown up that little tiny part, and we'll explain all the parts of an antibody a little later. But this part here is where the epitope actually attaches right through here. So this is a linear one, and this is a conformational one. It's bigger, okay? So linear are just like the individual like amino acids in there, so it's just that one single polypeptide chain, where the conformational is where we got folding of chains, you know, so it's more, like I said, a three-dimensional look to it. Now, remember we talked about the sizes, you know, of immunogen. It's got to be big enough. Well, sometimes we have some that are so small that we can't elicit an immune response. So they're considered non-immunogenic. And these are called haptens. In other words, they're too small for it to work. So here we're seeing a picture of it. So here that little red triangle is a hapten. And you can see it on a B cell. And it's not really allowing us to be big enough to elicit an immune response. So usually what will happen is you can see this little carrier here is taking the hapten. So that carrier, once it's combined with the hapten, will allow us to get an immune response. So here's showing you a hapten alone, where it's trying to attach, and we don't get the red cells, or excuse me, the antibodies to stick together. Now when the hapten is attached to a carrier, you can see where the antibodies will then attach, and they form like what they call agglutination. They clump together so you visually can see it. For example, poison ivy is a typical example that they use a lot of times, where it can actually bind to tissue proteins, and that's what gets you that red, itchy area on your skin, contact dermatitis. Other things that we have for immunology is called adjuvants. These are actually things they add to the vaccination or to the foreign thing to make it more immunogenic, to make it a better immune response stimulus. So we produce antibodies. You know, we want it to happen. You know, a lot of vaccines will add junk to them, substances to them, so when they give you the vaccination, it will elicit an immune response. So what it does is it actually stimulates the innate, the nonspecific, which actually will entice the more adaptive, the specific, to come in. So the adjuvant will make the antigen, this foreign thing, sort of like stay at the inoculation site a little bit better. so that we get a good innate response, which then will bring in the adaptive, more specific response. Because longer that substance is there, that immunogen is there, the more immune cells will be attracted to that actual site. So we get a better immune response. You've all had vaccinations. You get the shot in the arm, and you're like, God, my arm is sore. Well, duh, it's just because, and sometimes they can get red and irritated. That's because these adjuvants are there, and it's allowing the innate system to do an inflammatory response. I mean, that's what's going on. That's why it happens. But that's what you sort of want. You want people to have a little bit of a response there, so we get a better immune response for the patient. Okay, different relationships of the antigens to the host. We got different ones, and you just look at the prefix, auto antigen. In other words, that's what belongs to the host, like an autobiography. So it's antigens that are actually in the host. So if it is part of us, then we're not supposed to develop an immune response. I mean, we'll talk about it here in a second, but there are things on those autoantigens that will tell our body to not make an immune response to this. Now, unfortunately, our body sometimes can do this. And when that happens, that's when we start getting autoimmune disorders. So an immune response can happen to autoantigens. Alloantigens are from other members of the host species. Like when we talk about blood types, say if you're type A, that means you have A antigen on your cells, but you have antibodies to B. You talked about that in AMP2. So that means you've got alloantigens. Like some people are type B, so they have B antigen. Some people are type A, they have A antigen. So that's an alloantigen. Like I'm type A, somebody else can be type B, the different antigens. And this is just talking about blood types right now. But that's what we use as alloantigens. And they're very important, for example, what we were just talking about with blood transfusion. They're also huge in organ transplantation, like stem cell, kidneys, hearts, lungs, all that. We have to know what the alloantigens are of the patient to make sure the transplantation is successful and it doesn't elicit an immune response. There are heteroantigens, in other words, there's from other species such as animals, plants, or microorganisms. One of the ones I hear about a lot of time with microorganisms is group strep A. That comes into the body, excuse me, and your body will actually develop antibodies against this strep group A. And these antibodies will actually attack the valves of the heart and the glomerulus and the nephrons of your kidney. So that's a typical heteroantigen thing. That's why it's so important to identify group A strep for a sore throat. There's heterophile antigens. These are heteroantigens that exist in plants that are not related or animals, but they're very similar to what we have in our body. So an antibody to a certain heterophile antigen might cross-react with another one. It seems like sometimes we have antibodies, so like somebody develops mono, they can actually cause red cells from a guinea pig to agglutinate. I know it's like, who figured that out? But, I mean, so that's just a typical weird thing. Antigens from pneumococcal bacteria will react with type A blood antigens. So, I mean, it's like, it's a weird circumstance, but that's the kind of things that can happen. So that's what brings us to the MHC, the major histocompatibility complex. There is a minor histocompatibility complex also, but we'll just talk about the MHC. So MHC is important to determine, especially if we're going to be transplanting tissue, not blood per se, but actual tissue like stem cells, hearts, organs, kidneys, that kind of stuff. Because MHC is found on all nucleated, in other words, cells with a nucleus in your body, not red cells, but nucleated cells in the body. And they have to be compatible. And because we're talking about in tissue, that's why they call it histocompatible. Because if it's not compatible, these MHCs on the tissue, on the nucleated cells, then it'll be seen as foreign, so that means it'll be rejected. So that's why we do MHC typings on patients to determine what their MHC type is, so when we give organs that they're compatible. Now the MHC is actually linked genetically to you. So depending on what you have, you can elicit an immune response from that. In other words, just because your brother or sister, you might not have the same MHC, but if you'll remember when we do transplantation, they also look at, closely related like siblings and stuff like that. So it's the genetics that brings the MHC down to like the offspring and stuff like that. So the MHC is critical in the development of immunity, where we're talking about antibody formation immunity, which is humoral, or T-cell immunity, which is considered cellular. So these MHC complexes actually serve as carriers of protein antigens that can be recognized by the T-cells. So they actually look at it. So that's one way our body can sort of determine if it's us, like in other words, we're looking at our own cells, or are we looking at different cells? Because when we do blood transfusions, we're just giving red cells, and they are non-nucleated, so we don't worry about the MHC. Now, when you're giving stem cells, other organs, then you're talking about cells with a nucleus. So that's where this MHC plays a critical role, and that's why the T cells will recognize these if they're foreign and will develop an immune response. So this genetic coding of MHC is actually found on chromosome 6. There's actually 3 different categories: class 1, class 2, class 3. Those 3 categories, as you can see listed here, class 1 is on 3 different loci. So it's A, B, and C. Class 2 is found in the D region, and it's called DR, DQ, DP. And class 3 is actually between 1 and 2, which I don't know why they came up the way they did it, but whatever. And it codes for complement. That's why you hear it C4A, C4B, C2, and B. And when we talk about complement, we'll hit those topics again there. So MHC, there's class 1, class 2, class 3. And so when you see HLA typing that we'll talk about here in a little bit, you'll see where this comes into play. So this is showing you the chromosome 6, and we're not going to ask you this on a test, but I just want you to see, there's the class 1, here's the class 2 that we were talking about, the DP, DQ, DR, and class 3 here. This is basically complement in there. So when you're talking about MHC, we're talking mostly about, like we said, nucleated cells. So a lot of times you'll hear people talk about it, especially when we're talking about blood kind of junk. We're talking about the white cells. So HLA actually stands for a human leukocyte antigen. So that's really another way to say MHC. And the HLA system is very polymorphic gene system in the human. In other words, it takes on all kinds of stuff. There are millions, it says 1,000, but there's really millions of things out there. So there can be all kinds of alternate forms of this gene, and it can be, different varieties on the same product. I mean, you can see here it's saying there's 4,600 different HLA just A's, 5,500 HLA B's, and 4,300 HLA C's. So every person has 2 HLA alleles of each type. And this is actually inherited as part of your haplotype. You inherit it one from each parent. Okay, that's why there's 2 HLA alleles for each type. So class 1 are on all nucleated cells, not red cells, but all nucleated cells. They differ in how they're actually expressed on there depending on what type of cell it is. So we see them on highest amounts in lymphocytes and myeloid. That's why it's, we do HLA typing on actual blood samples. And they're lowest like in liver, nerve, sperm, what else? Muscle cells. So that's why when we do HLA typing or MHC typing, We're doing synonymous here. That's why we look for it in blood cells, particularly white blood cells. So here's showing you a class 1 MHC molecule here. So you can see it looking at it, the class 1 molecule is a glycoprotein dimer, and it's got 2 non-covalently linked polypeptides. There is the alpha, you see alpha 1, alpha 2, alpha 3 here. That's 3 different domains there. And then off to the side is a beta 2 microglobulin. That's actually a different chromosome there. But you can see how it's bound. You know, this is a cell wall here. So you can see how it embeds itself in through the cell wall of it. And remember, these are in nucleated cells. So class 1 MHC molecules process antigens endogenously. In other words, it brings it in. And we'll go through the steps here in a second. And presents them to a T cell, and particularly a CD8 cytotoxic T cell. All right. So here's showing you the T cell here. Okay. The CD8 cell. So this endogenous antigen that is within the cell, the cytosol, is degraded by a proteasome. So these peptides will transport into the endoplasmic reticulum. Okay, so here's a proteasome, the protein, and due to TAP, which is, and I can't remember what it stands for all of a sudden, but TAP, it will get inside the endoplasmic reticulum. Okay, so once we're in the endoplasmic reticulum, remember here's the alpha, the little green, there's the beta 2 there. So the alpha class 1 binds to the peptide. So you see it, the peptide there. Okay, so once it gets into that peptide, the alpha chain, it will then move on through here to the Golgi complex, you know, inside the cell itself, transport it to the Golgi complex, then goes to the cell surface so that it is now exposed so the T cell can then react to it. So you see how the CD8 has this little external thing. It's looking at the alpha chains here, but what it's looking at right there, which you can see there's the receptor there, that's where it binds, that little endogenous protein that we brought inside of the cell in step 1 is actually attached to the MHC, so the T cell sees it, and so now it knows to react with it. So that's what it means by endogenous. It's got to go through the cell so that T cells will be presented to them so they can react to it. So once the T cell reacts to it, you know, and it's like, oh my God, there's something foreign there. I don't like that. You know, so what it will do is it release its, because it's a cytotoxic cell, it'll release its stuff into that, whatever foreign thing it is, and destroy that target cell. Apoptosis, it destroys it. Alright? Now, that was class 1. Class 2 MHCs are on antigen-presenting cells. And if you remember from the previous chapter, we said antigen-presenting cells are B lymphs, monos, macrophages, dendritic, and particularly thymic epithelial cells, you know, T cell stuff. So remember, class 2, we said, is DP, DQ, and DR. So they look a little different than we said in class 1. They have 2 non-covalently bound polypeptides that are encoded by separate genes from the MHC. So you can see alpha 1, alpha 2, beta 1, beta 2, and you can see how the peptides interlock up here. Okay, so this is the peptide bonding cleft, but you can see they have 2 things going inside of that cell membrane. So the alpha chain has 2 domains, beta chain has 2 domains to it also. So the class 2 molecules do it more exogenous. Instead of endogenous, it's exogenous antigens, and it will present to the CD4 cells. So here's class 1, you know, or class 2, excuse me, step 1 at the bottom here. So the class 2 MHC binds to the chain to block to block binding of endogenous stuff. Okay, so the MHC complex, so you can see that little link is an invariant chain, so that makes it class 2 because we've got the 2 alpha, the 2 beta there. It goes from the ribosome to the Golgi complex. And again, we have no foreign thing on there like we did with class 1, like the exogenous one. Okay, so the exogenous antigen, so we go to step 3 here. So it has this, they call it an invariant chain. It's actually called a clip, is what the actual fancy term is for it. So that clip fragment will come up here, and it's actually starting to degrade here, but it leaves that little yellow square, we'll say, up there. So it goes up here, and there you can see that here's the antigen coming in. So when we go into step 4, here's where it goes inside the the vesicle inside of the cell, which is, into the cytoplasm here. And then in step 5, that little clip fragment, that little white or yellow square there, you see, will be exchanged for the foreign thing. So if you go to 5 to 6, see that now that yellow thing is now that foreign thing. That little antigen is now attached to that there. Right? So now, in step 6, that'll be pushed to the cell surface, and when it's pushed to the cell surface, the T helper cell with his little stuff will attach to the alpha, and then the receptor part right there, the T helper cell will bind to that complex on there. So class 1 is endogenous antigen processing. Class 2 is exogenous antigen processing. Class 1 is CD8. Class 2 is CD4. And that plays an important part when we talk about the immune system a little bit later. Now, the T helper cells, the CD4 cells, will actually, they stimulate B cells to produce plasma cells. So here, remember, this is the antigen presenting cell, and it's given into the T helper. This is from the previous slide. So here, we're at having the exogenous thing here, the MHC2, the exogenous, the T helper cell. Well, it squirts cytokines out, one part of it, and then it makes more T helper cells there to proliferate. But let's get over to the cytokine secretion of the T helper cell. It now tells the B cell, hey, B cell, you need to transform into these purple things at the bottom, plasma cells, so you can start making this Y mess. This Y stuff down here is actually antibodies. Because the B cell will not know what to do until the T helper cell has got that exogenous antigen attached to it and the cytokine release to tell it that information to make that little blue square foreign antigen there to make antibodies to it. That's how it goes through it. So there's a lot of processes that got to go on. Now, let's look at the difference between class 1 and class 2. Okay, class 1 is all, repeat, all nucleated cells. Class 2 is the antigen presenting cells that we listed. Class 1 is 1 alpha chain, remember it had 3 parts, and the beta 2. Class 2 is an alpha and a beta domain. Class 1 is A, B, and C. Class 2 is DP, DQ, DR. Class 1 is a little bit smaller amount of amino acids that'll bind. Class 2 is a little bit bigger. The peptide bonding cleft is closed here on 1, but class 2, remember, it was open because that's how we did the exogenous antigens there. And then class 1 uses CD8 as an endogenous antigen. Class 2 uses CD4 T cells for exogenous, so endogenous, exogenous. So what's the clinical significance? Why the hell have all this MHC stuff to begin with? Well, it's important, because this is how we determine if it's compatible or not. That's what we're looking at. So it's very important in tissue transplantation. So they want to make sure when we're giving tissues to people like stem cells or organs that the class 1 and class 2 MHC will be compatible. Because if it isn't compatible, then we'll induce a graft rejection. So what we do is we actually type them for MHC. It will actually be called HLA is what it will actually be called. Because we're looking at white cells. Because remember we said white cells in particular have more MHC on them. So they're a better way for us to get the information we need. So that's why we use human leukocyte antigens, HLA, to do the typing. So that's important to know what the HLA typing is when you're doing organ transplant. Not blood cell, red blood cell donor blood, red cell transfusion, not that. Because there's no nucleus in there. But we have found out that certain HLA types seem to play a huge role in certain autoimmune disorders. Because remember, we're supposed to be able to see it's ourself, and we're not supposed to elicit an immune response to ourself. So it seems like certain HLA types, we will elicit a certain immune response to it. So here's a few of them here. You can see ankylosing spondylitis. That's where you have inflammation in the spine, all the vertebrae, and you can see it's HLA-B27. Celiac disease, DQ2, DQ8. Rheumatoid arthritis, DR4. And type 1 diabetes is DQ8 and DQ2. I mean, it's interesting. A lot of times you won't see HLA typing done to determine celiac or rheumatoid or type 1 diabetes. I mean, there's so much other tests that are done. Ankylosing spondylitis, they will check for HLA-B27 to make sure it's there. So, and you can see this association is very strong for HLA-B27 for ankylosing spondylitis. So in summary, immunogens, the bigger, the better. They'll elicit a better immune response. So it makes us produce antibodies. Another word for antibodies, as you see here, is called immunoglobulins. That's another fancy word for antibodies. Or it can make the lymphocytes become sensitized. I mean, it's what we were just talking about with MHC class 1, class 2. Remember the endogenous, exogenous. So that's called sensitization of lymphocytes. And people that can elicit an immune response. That's called an immunocompetent person, somebody that can produce an immune response. If you can't, then you're called immunosuppressed. An antigen, it just means it's foreign. It doesn't mean it can produce an immune response in a host, but it will react to an antibody once it's formed. And when we start doing, testing, you'll see this. Immunogenicity, in other words, can we produce antibodies or not? That's affected by your age. Remember, young and real old are not that well. Health, stress, fatigue, malnutrition, and your genetic makeup. The heavier the molecular weight, the better. The more complex, the better. And depending on the route of inoculation, the better the immune response. Epitope is actually what's on the antigen, the foreign thing. That's actually the antigenic determinant site. That's another name for it. That's what we use to recognize it being foreign. So the epitope is on that immunogen, and that is what is recognized by the B and or T cells. Haptens, or in other words, are just too small. They don't fit the qualifications we need to produce all by themselves to produce an immune response. So they're considered very small, non-immunogenic. But they have to be bound with a carrier, something bigger, to produce an immune response. And a lot of times, if we mix an antigen with an adjuvant, that will elicit a better immune response to it. Look at vaccines that you get. Look at the contents of it, and you'll see adjuvant and it'll list what it is. They're usually a salt of some sort, that will enhance the immune response. I mean, you've had shots. Like we said, you can feel it. Autoantigens, in other words, that's our own antigens. Alloantigens, same species, but comes from a different person. We were talking like blood types, like type A, and type B. I mean, we're still humans, but certain people have A antigens for type A blood, B antigens for type B blood, for example. Heteroantigens, they come from other species. Heterophile antigens, they're unrelated, but they seem to cross-react different species. I mean, it's really weird how it works. And we take it to our advantages a lot of times when we're doing testing. MHC, we have class 1, class 2, and class 3. Class 3 we really didn't talk about that much. That's more complement stuff. Cytokines also. But class 1 and class 2 play a huge role in how we present antigens to T cells. So class 1 and class 2 actually bind to certain proteins, peptides on the antigen within the host cell, and depending on if it's class 1, we do endogenous presentation, or class 2 exogenous presentation to the particular T cell. Remember, class 1 was 8 T8s, cytotoxic, and class 2 was helper cells, CD4 cells. So, well, that's what I just said. Class 1 presents endogenous antigens to CD8 trigger and cytotoxic reaction. Class 2 is exogenous, using CD4 cells, which then make them produce antibody production. So HLA, very polymorphic. In other words, you get a whole bunch of different types with that. Thousands of them out there. I mean, when you get it all together, there's a bunch of them. And we just showed you that certain HLA types are associated with the certain autoimmune disorders that are out there, like the HLA-B27 ankylosing spondylitis. Alright, so that's it for Chapter 3. So make sure you go through the textbook. We went and showed you a lot of the pictures. Read the summary, and you can see the study guide that's back there also. And then it has a couple case studies and review questions, so you can go through those. And like we said, the answers in the back of the book. Very important stuff. You got to understand it all. I mean, we are building on immunology. There'll be more and more all the time, but you got to understand this before we get to the next step. So if you have any questions, make sure you ask me, email, text, whatever. Talk to me in lab. Okay, that's it. Thanks.